Hello from Tanima. You're watching this video in my YouTube handle at @Tanima_Barbie. Just to let you know, my YouTube venture is totally a personal, anywhere is a voluntary virtual social outreach, and it has nothing to do with my profession that engage with research and teaching in the field of molecular biology and biotechnology in an institution in India. I personally believe that education should be freely accessible to all, and with this in mind, I have initiated a series on cancer biology. This is my 44th video on cancer biology. The present video intends to discuss on few perspectives of metals in mutagenic mechanisms of classical carcinogens in the topic on environmental exposure and cancer connection. The present video partially discusses the paper entitled "Not All Carcinogens Are Directly Mutagenic, a holistic view of the environmental exposure cancer connection." The DOI of the same has been pasted in the description box. I would also request you to turn on the subtitle or caption button on your screen in case you find difficulty with my deliberation. Do keep watching. In contrast to nickel, cobalt was among the limited number of agents that exhibited a distinct genomic signature in tumors derived from metal-exposed mice. It was linked to an increased frequency of KRAS mutations at codon 61 when compared to other chemicals, which tended to affect codons 12 and 13. Lung tumors in mice exposed to cobalt also exhibited an increased frequency of dinucleotide substitutions. Moreover, the mutational signature observed in the lung was different from that derived from the liver, indicating that tissue-specific factors could modify mutational damage or influence how a metal interacts with cells from that particular tissue. Conversely, the biological impacts of cadmium, classified as Group 1 carcinogen by the International Agency for Research on Cancer, or IARC, seem to be more complex. Cadmium transformed prostate epithelial cells exhibit a gradual rise in DNA methyltransferase 3-beta or DNMT3B expression, indicating increased hypermethylation, while showing no alteration in DNA methyltransferase 1 or DNMT1, indicating no impact on hypomethylation. Elevated levels of DNMT3B were associated with a decrease in the expression of tumor suppressor genes RASSF1A and P16. Supportingly, cadmium has been demonstrated to facilitate malignant transformation in previously non-cancerous epithelia via the ZIC2 and GLI1 pathways. These assays were conducted using systems that monitor the behavior of a bulk cell population, rendering any mutagenic effect as a contributing factor to the phenotype improbable. However, mutagenic roles for cadmium have been documented. So viewers, hope the video could discuss few perspectives of metals in mutagenic mechanisms of classical carcinogens. Please don't forget to like and share the video and also subscribe to my channel. Here I would like to mention that the references that were used for preparing the video have been pasted in the description box, and if you're interested to look into the details, you might check the same. Hope to meet you soon with yet another video on cancer biology. Till then, do take care. Keep well.